In this goal, we are going to be using our knowledge of intermolecular forces and applying it to the shape and function of large molecules. On the AP test, they often give you molecules that you've never seen before. And so the purpose of this goal is to help you look at the large molecule and break down what's there so you can make a decision about the bonding that occurs throughout the whole molecule. Let's first start off with protein. And in protein, it's composed of amino acids. An amino acid is composed of something that has an amino group on the end, this NH3, and then a carboxylic acid group on the end. And through the process of a reaction, you get what's called an amino acid. If you take many of these amino acids and you link them together, then that is the start of your protein formation with peptide bonds. So these large molecules, their shape equals their function. So in other words, how their shape is how they function in an organism. And large molecules get their shape through intermolecular forces and with folding and curling up on itself. So the amino acids are represented here by a little circle, and they are linked together with peptide bonds. And this is what's called the primary structure. And so this is what it looks like. The amino acid chain will fold up in one of two ways. It can fold up with an alpha helix right here, or with what's called a beta pleated sheet. And how it does this is by linking throughout the long skinny molecule where it will hydrogen bond. So through hydrogen bonds, it's going to either link up as a spiral, so kind of like an old-fashioned telephone cord will spiral up. So that's how this will wrap up. Or it can do this pleated sheet. And again, it's all about the hydrogen bonding. It can fold even further, and when it does this, that is called the tertiary structure, where it folds up even more. And again, it's because of hydrogen bonding, and you also might have what's called a sulfur bond there. And this right here, this quaternary structure, this is showing 3 folded up amino acids that are linked together. And now this is maybe an enzyme that can go and do something in the organism. But how it is wrapped up determines how it will react in that organism. And it all has to do with hydrogen bonding. You're going to find most of these large molecules, they all have to do with hydrogen bonding. We briefly talked about DNA before and how there is hydrogen bonding that is linking the base pairs together. Those base pairs are given letters G, C, A, and T. And those little dots right here, that is the hydrogen bonding. Here's another picture of that we've gone through. So again, we have hydrogen bonding that's linking one base pair to the other in that double helix of the DNA. Oil and water, do they mix? Let's look at a little simulation. So we have water that's represented as the plus and the minus. And just for simplicity's sake, they just have 2 atoms there, even though we know that water has 3 atoms involved. And then we have oil, which is a very, very long, skinny molecule that is nonpolar. And so that's why you don't see the plus and minus on the oil. And we're going to let this simulation run. So we're going to shake up the oil and water mixture. And we're going to see just with the collisions that the polar molecules, they are going to be attracted to each other, and so they will start to clump together. And you will see the nonpolar molecules, they will get isolated out. They all have dispersion forces, but you also have a stronger dipole-dipole and hydrogen bonding between the water molecules. And so you are going to see them clump together because there are stronger interactions with water molecule to water molecule. So eventually you will see that they will be separated. And density also has something to do with it. Oil typically is lighter than water, and so you'll see the nonpolar molecules clump together at the top of the water. So do oil and water mix? And the answer is you can mix them up, but they won't stay mixed. And that is because of differences in density and also their intermolecular forces. The polar is just going to be very attracted to polar. Soaps, why do they work? So if we look at a soap molecule, it has a very, very long carbon chain. And remember the carbon-hydrogen bond that is nonpolar. And so if you put a whole bunch together, this whole portion of the molecule is going to be nonpolar. And this portion of the molecule on the end, this is going to be polar. So when you get a dirt particle or a grease particle, it's going to do different things. So your grease particle right here is nonpolar, and so you're going to see it being attracted to the long skinny part of the soap molecule. And so you can see that the grease particle is going to be surrounded by the nonpolar tail portion, and then you have water on the outside, which is polar. And what this does is those non-polar ends of the soap, they're able to grab onto the grease, and then you can use water to rinse it away. Soap is great for grease, but it's also great for dirt. The dirt particles, they will be more attracted to the polar ends. And again, the grease particles, they will be more attracted to the non-polar ends. And so this is how soaps work to get out grease and get out dirt. This is a phospholipid, and it is important in cell membrane. And we have a ball and stick model, and then we have a model that just shows the individual atoms. But from this, you should be able to tell me where is it nonpolar and where is it polar. And you would see that in this area, this would be nonpolar. In biology, we call this hydrophobic, and that means water hating. And so nonpolar portion will hate water. This portion over here, if you look at it, you have all these little charges and you have these oxygens hanging out with their unbonded electron pairs. And then here's some more charges over here. So what's happening at this head is this is polar. So a biology person would call it hydrophilic, meaning water loving. So when you take a whole bunch of these phospholipids and put them together, you get a cell membrane. And in your homework, you need to sketch a cell membrane. So to sketch this, just make your life easy. Do the hydrophilic heads and the hydrophobic tails. And so you'll just sketch them on one side, and then you'll put its opposite neighbor on the other side. So what we end up getting is in the middle, we get all the nonpolar tails, and then we get all the polar heads together. And it's because of their attractive forces. So we're going to get dipole-dipole here, and perhaps hydrogen bonding, depending on what's there. And we're going to get only dispersion forces here. Of course, there's dispersion forces in the hydrophilic heads too. So sketch out this little cell membrane just as a representation of how these big molecules react. Again, polar will go with polar and nonpolar will go with nonpolar. Another large molecule that you might see on the AP test are polymers. And polymers are long chains of a monomer or a base unit. And you can tell it's a base unit because they'll use these brackets. Here they're using these parentheses. So this is a repeating pattern over and over, and these end up being really, really long, skinny molecules. Some examples of polymers are starch, protein, DNA, which we've already talked about. It is considered a polymer. Rubber, silk, plastic. I have a plastic down here to show you. Nylon, styrofoam, Teflon. I do have Teflon down here to show you. You should be able to look at the polymer and look at its base monomer and figure out, is this polar, is this nonpolar, and therefore how will it react. So let's first look at our plastic. So in our plastic, we are taking a repeating unit of what is just carbon and hydrogen. And so what do we know about the carbon-hydrogen bond? We know that it's nonpolar. So if you have a whole bunch of these all together, we get this plastic polyethylene, and it is nonpolar.